Hi everyone, my name is Mariangela Pappadà and I work as a scholarship holder at the Department of Chemical and Pharmaceutical Sciences of the University of Ferrara, Italy. Our research focuses on the development of a therapeutic strategy for the treatment of Spinocerebellar Ataxia type 1. This project points at the permanent silencing of the endogenous ataxin-1 genes, with the aim to obtain the block of the production of toxic protein. For this purpose, our research group used the CRISPR-Cas9 system, consisting of a guide RNA able to bind and form a complex with the Cas9 protein that is guided to the target DNA by the presence of a complementary sequence. Once produced, the ribonucleoprotein complex must be introduced into cells, where after bonding Cas9 generates a double-strand break with flat ends, with the subsequent activation of the double helix damage repair mechanism, called non-homologous enjoining, that leads to gene silencing. We designed 10 different sgRNAs able to recognize sequences upstream and downstream the ataxin-1 poly-Q tract, and tested the efficiency to recognize and mediate the cutting by Cas9 of these sgRNAs in vitro reactions. The efficiencies were determined as a percentage of the fragments obtained from the Cas9 cut related to the total amount of the target sequence. At least 4 sgRNAs show a high capacity to mediate the cut of the target sequence, 2 of which recognize sequences upstream and 2 downstream of the poly-Q tract. Since a multiple cut of the ataxin-1 gene can determine loss of genetic material and therefore a more efficient silencing of the gene itself, we set up 4 pairs of sgRNAs, whose recognition and cutting efficiency was always very high. From preliminary tests on immortalized cell lines, we chose the G3-G8 sgRNA pair, which confirmed the ability to effectively knock out the ataxin-1 gene expression. As there are no cellular models for SCA1, we decided to use fibroblasts isolated from 5 patients with different poly-Q expansion in mutated ataxin-1 gene. Fibroblasts were then transfected with G3 and G8 sgRNA simultaneously and with a negative control sgRNA as nucleofection control, previously associated with Cas9 to obtain ribonucleoproteins. Western blotting analysis with anti-ataxin-1 antibody shows significant reduction of the ataxin-1 expression from 27% to 75% of expression compared to not treated cells. To verify which genetic alteration were introduced by our CRISPR-Cas9 system, we decided to amplify the region of exon 8 containing the poly-Q and the sequences recognized by G3 and G8 of the treated cells of 3 patients, and to subclone PCR products. Following sequencing and alignments with the correct exon sequence, we were able to determine in what percentage indels and point mutation were introduced or the poly-Q was completely excised, obtaining 13% for indels, 14% for point mutations, and 2% for poly-Q deletions. G3 and G8 sgRNAs mediate the excision of the poly-Q tract only very few times, only 5% in SCA1 and 8, probably due to the steric hindrance caused by the nearness of the target sequences and the high molecular weight of the Cas9. G3 sgRNA show a higher efficacy with a ratio of about 3:1, and the largest number of indels and point mutation were localized upstream of the poly-Q tract compared to the cut mediated by G8 sgRNA downstream of the poly-Q. In conclusion, in this study we demonstrate to successfully reduce protein expression from SCA1 patient-specific cells using the CRISPR-Cas9 system, obtaining suppression efficiencies that agree with those obtained with antisense oligonucleotides, but with the advantage of permanent suppression without the need of continuous administration. However, this result will be confirmed in SCA1 fibroblasts of other patients that we have already recruited. Before leaving, I'd like to mention Professor Peggy Marconi and Dr. Francesca Salvatori, who work with me on this research project, thanks to the support of foundations such as ACAREF and ISA at the forefront of research on ataxic syndromes. Thank you for your attention.